So our next speaker is Swami Venkatraman, and he's at Penn State and at Rice University. He's an adjunct professor, and please go ahead, Swami.
Okay. All right. You can all hear me well? Okay, good.
Yes.
Thank you. Thanks, Mauricio, and thanks the Carbon Society for giving me this opportunity to talk in the special COVID-19 meeting. I just want to start with a disclaimer. I'm neither a virologist nor an epidemiologist. And the reason I say this is because unlike the other speakers, I had the honor of sharing this audience, like Professor Kuchipudi, I'm not a virologist. And it's an honor to be in the same forum as the inventor of the mask, the surgical mask, and the other speakers who talked very eloquently. What I have done is, I was fascinated for two reasons. One, of course, this is a very unforeseen pandemic of a great magnitude, almost close to 500,000 deaths so far, but also the curiosity that, like everybody else, that I have about this virus and how it operates and so on. So it made me to sort of learn about this. The purpose of sharing that learning with you is to see what opportunities as a material scientist one can have in not only doing something for the current pandemic, but really more importantly, what we can do for the future pandemic. So my topics, I have arranged it in these various areas. Since many speakers have already talked about it, I can sort of go through them quickly about the COVID-19, the disease, and the virus, which is the SARS-CoV-2. Since some new people have joined this afternoon, so I'll probably go through them, even though they have been talked about previously. And one area that I want to stress a little bit is the testing aspect of this virus and the disease. How there is a lot of still unknowns surrounding this testing. Professor Kuchipudi talked about the transmission, and I will also share with you some data based on why this particular virus is so difficult to manage. And then the environmental stability, and the previous speakers also talked about this, but again, in the context of what future opportunities could exist, I will talk about it. Sensing, I will talk about some new ideas. Professor Zhang very eloquently spoke about the carbon FETs, graphene and carbon nanotube-based FETs. But this topic is also very important in terms of be prepared for future. Whether we can start looking at new technologies, new innovations in terms of preparing antiviral approaches, and then summarize my talk. About the disease, the COVID-19 is the disease. Professor Kuchipudi already showed this slide, so I don't need to belabor this point, but it's a pandemic which has not been anything like what has been seen before, at least in the recent times. So already close to more than 500,000 global deaths. And if you look at these two charts here, presenting that information slightly differently. First of all, what is the contagiousness of this COVID-19 and how deadly it is? And one way to look at this is to plot the infectiousness of the how many people die, the deadliness of the disease as a function of the contagiousness, which is described by this R0 parameter. That is the average number of new infections that a single infected person can cause. And if you look at in the context, the COVID-19 occupies somewhere around same R0 factor, like even common cold, swine flu, seasonal flu, etc. And in terms of the deadliness, and as previously pointed out, it's not as deadly as the SARS or the MERS, which are the typical, or the other two corona-based virus infections. But nevertheless, the infectiousness is, it varies around 1.5 to 3.5, and the fatality rate also varies. But on the right side, if you look at the number of deaths per day, this is updated last week, 7th of July. And again, compared to the rest of the devices, the SARS-1, when it was incident, it was contained very quickly. The MERS also contained very quickly, but compared to the swine flu, HIV, AIDS, and then you look at the corona, it is very closely resembling another major pandemic in the world, namely the tuberculosis. So in other words, the disease is something that is, even though the numbers are similar to what has been seen before for some of the commonly known viruses, but nevertheless, the disease the deadliness factor is fairly high. And why is it so? And again, it's also important to keep in mind that this R0, the subscript, refers to the early stages of the epidemic, and so that doesn't necessarily say what that number is or how the disease spreads when certain preventive measures are already taken, such as wearing a mask and so forth. Now, if you look at what is the reason for the rapid spread of this COVID-19, again, I just want to remind you of my disclaimer, but my interpretation of this is just based on what I have assembled from the literature. The question which is important to ask, and at least that's where I start off, is what is the dose for this virus for infection? Before somebody say that an infection has happened, what should be the dose? And again, there's not really much data, but based on some study on ferrets are like, again, belonging to the cat-like animals, the TCID, which is the median value of for an infectious disease to occur, the TCID 50 number, it's roughly around 467 for nasal washes and about 53 for saliva. How does this number compare? That is the question to ask. And if it compares again, if you compare it against the common cold, then the onset of infection, the TCID number, is really very small compared to an orders of magnitude small compared to what could be the dose that is required, dose of the virus that is required for the infection. So this is one factor. And Professor Kuchibudi mentioned this a little bit. The coronavirus, both the CoV-1 as well as CoV-2 have a very long incubation period, meaning the time from the infection to the onset of the system than the influenza viruses. So the mean incubation period for CoV-1 is roughly about a week or so, but the CoV-2 is roughly of the order of a couple of weeks. And on the other hand, the influenza 2009 is of the order of about 2 days. So the incubation period is longer for the CoV-2. Now, the next point is also very important, and that is the average viral load during the time that a person is infected. In other words, during this incubation period, again, it's a fairly high number, close to about 1 million copies per upper respiratory tract swab in the first 5 days. And in this, there is not much of a difference in the infectivity of symptomatic and asymptomatic persons. And on the other hand, if you look at CoV-1, the viral load peaked after the onset of symptoms, that is 6 to 11 days after the onset of symptoms. So what this high viral load in the first time of incidence, what that means is it's very hard really to mark the isolation and then the quarantine of patients and their contact. So this makes this isolation and quarantine aspect of this disease very, very difficult. And that's one of the reasons perhaps is that it has spread to the extent that it spread. Then the next bullet is also critical. The viral shedding seems to also occur for this CoV-2 over prolonged periods of time through the swabs, sputum, and stool, etc. Actually, it lasts for a couple of tenths of days. So basically, the coronavirus 2 in comparison to the coronavirus 1 or the MERS viruses, the previous type of coronaviruses, the clinical characteristics are very different. And the understanding of this is also critical in terms of containing the spread and then also coming up with the preventive measures. The previous speakers again talked about the structure of the virus itself, so I can quickly go through it. The SARS-CoV-2, Professor Kuchibudi mentioned, it's one of 7 coronaviruses. There are, of course, the CoV-1, the SARS-1, the MERS, which is the Middle Eastern Respiratory Syndrome virus, but there are 4 other types of viruses which are more like the common cold type of viruses. And this is not a naked virus, it's an enveloped virus, meaning it has a lipoprotein coverage, and it is a class 4 and a class 4 single-stranded RNA, that is an mRNA, which simply means that it doesn't require a DNA for its reproduction thing. And so it's a large-sized virus, about typically varies from 80 to 120 nanometers. And also the fact is apparently it is the largest mature RNA virus. So this is larger than many other viruses known to us. And the nucleocapsid, the protein which envelops the genome, for a class 4 type, it has a helical symmetry, while the class 4 usually will have an icosahedral symmetry, a symmetry with 20 triangles arranged. So this helical symmetry is also some uniqueness of this virus. And again, what consequences it would have in terms of how the virus is destroyed or how the vaccines are developed and so on, I don't know, but I would probably speculate that it might have some importance. And the other speakers also talked about this. How does it bind? The spike protein, there are about 100 of them, 100 copies of them from a virus particle, and the length is roughly about 10 nanometers. In addition to the spike protein, you have the envelope protein and the matrix, the membrane protein, but these seem to be the largest number, these spikes, and the spike protein then binds to the ACE2 enzyme, and from this then it attaches to the host cell and then multiplies. And this diagram just shows how that process happens. The way the spike protein enters the human cell also seems to be somewhat interesting, or at least the way I have been trying to understand this. It seems to take place in 2 functional domains. First of all, it has to bind to a receptor on the cell, so that means there's a receptor binding domain. Then there's a second domain. Once the receptor is bound, how it cleaves and then enters into the cell, cleaves and enters through the cell membrane. So therefore, this seems to be an interesting way this the ACE2, which is the angiotensin converting enzyme 2, seems to be interacting with this spike protein. And also another enzyme which breaks down protein seems to also be playing a role, the temperase 2, the protease enzyme. And that could be playing a role in terms of how the membrane, once it is broken, how the virus enters into the body, into the cell. The reason why this coronavirus and the disease, the COVID-19, affected various kinds of people in different types of population, different underlying symptoms and so on, because perhaps of the fact that this enzyme, the ACE2, is in many cell types. It is in the tissues, including the lungs, the heart, blood vessels, kidneys, liver, and gastrointestinal tract. And actually some of the symptoms, if you look at the symptoms of COVID-19, symptoms associated with all these body parts were seen. Such things like diarrhea and so forth. Some people developed blood clots, and then subsequently that led to fatality and so on. So in terms of therapeutic and vaccine, Professor Kuchibudi also already talked about it. The responsible enzymes, how you attack the enzymes, the potential targets for antiviral intervention, and the treatment with the anti-ACE2 antibodies disrupt the interaction between the virus and the receptor. Now, in terms of testing, the SARS-CoV-2 testing, generally, there could be a direct test. You can culture the virus and then you can look under the microscope. But generally what is used is the non-culturable by detecting the pathogen antigen, by using looking at the AGs, the antigens that are present on the cell wall of the pathogen, and then use primers to bind it and then amplify it. And then you do the nucleic acid amplification test. And for the DNA viruses, you use the polymerase chain reaction, but this being the single strand RNA virus, you use the reverse transcript PCR. What that means is it converts the RNA to a complementary DNA and then detects it. The other way of detection is detecting the antibodies that are developed if the symptoms are there. And so therefore there is serological tests are done. And of course, this is naturally, the symptoms should already be manifesting. So it's a retrospective testing. And you look for primarily these two antibodies, the IgM and the IgG, which develop against the host surface antigen or against the toxins produced by them. So typically the PCR is, the RT-PCR in fact, is one of the major techniques that are used for testing. Now, what is important when we do testing of anything for that matter, and especially now in the context of the virus testing, we have to understand what is known as the receiver operating characteristics. What that simply means is you not only be able to identify the true positive and identify the true negatives, but every test has deficiencies. They are not completely 100% positive or 100% negative. So you have, in addition, false positives and false negatives. So we need to get a handle on what is the positive predictive value and the negative predictive value. So I have shown here, just for illustration only, this what is known as the receiver operating curve, where you really plot against the true positive versus the false positive rate, and if everything is good, you get the straight line for a random guess, but that's not really what is ideal situation. You want a perfect classification where you have 100% true positive and the false positive is zero. So if you look at the various sensors, here are some examples that are given, is purely illustration. So there are 4 types of sensors, let's say, with these values of true positive and false positive with these values of accuracy. You really want a sensor which is better, which means it has to be on the upper side of this dividing line, and anything that falls below is not really good. This is an important fact, and I find, looking at the literature, that this is something which is always overlooked when we report a new sensor, that it can detect so and so, or it can detect this much of sensitivity, and so on. But then we forget what is its operating characteristics, especially under the clinical condition. So I have shown here a result based on a published result, published in around February 2020. This is when the disease is sort of taking off, and tests done over 500 and over about 600 people. This is actually the CT scan of the lungs, which Professor Kuchipudi talked about, looking at the opacity in the lungs, and then the reverse transcript PCR for actual virus determination. And looking at these 600 so cases, if you look at even when both are combined together, even though the sensitivity is very high, that means if it is somebody is positive, then you test that person to be positive. But if somebody is negative, you don't really test the true negative, the specificity, which is the specificity. It's abysmally is very poor. It's only around 25% or so for a 95% confidence interval. So you can see how the testing that is done for this identification of this virus can be a little bit of a problem. Now, Another data that I want to share with you, again, published data. Again, combination of CT and RT-PCR, in this case, around 51 patients. Out of the 51 patients, only 35 out of 51 patients, when they tested positive with one, namely RT-PCR, they tested positive with the CT at the initial presentation. That means this is the sensitivity number, even for this combined result, is only about 70%. So it's not really very high. Now, there's much talk about this molecular test, which is by Abbott Labs, this Abbott ID Now. From their website, they claim 91% sensitivity and a specificity of about 100%, but the jury is still out. It is presumably a low-cost device. This number is taken from news press. It is not published on their website. Roughly about, say, 5K. So considered to be a cheap or a low-cost testing, and they claim these numbers to be very high, but that is still, as I said, the jury is still out on this. So, so based on the testing where the receiver operating characteristics are not really the greatest, still that's all we have. So now we look at the transmission part of the virus and how the disease spreads. I mentioned already the high viral load on presentation. If you look at in comparison to CoV-1 and CoV-2, from two samples, throat versus saliva, and these numbers vary quite a bit. In the sense it can be anywhere from 1,000 copies per milliliter to roughly million. But in terms of the COVID-2, you can see in the throat specimen, it's much, much higher, and the saliva is about comparable to CoV-1. So what that means is the high viral load on presentation suggests that the virus can be transmitted asymptotically or pre-symptomatically even before the symptoms develop. And how does it happen? Through coarse and fine droplets generated when the patient talks, coughs, or sneezes. The patient meaning here somebody who has this kind of a viral load. So then how does it spread? Okay, this high viral load and a person coughs or sneezes or whatever. So if you look at the aerosol infection dose, how many virus particles in the aerosol? Again, if you look at comparing CoV-2 versus an influenza after 1 hour of exposure, the copies of the virus particles can be a few thousand, and the CoV-2 is also of the same order of magnitude. And the infection is proportional to the exposure to the virus and the time of exposure. And this is some interesting statistics that I want to share with you, and why, as somebody said earlier, why wearing a mask could be important. A cough releases close to 3,000 droplets, and these droplets travel at 50 miles per hour. And so they can very easily travel across your room. On the other hand, if somebody sneezes, it's about 30,000 droplets, and they travel very fast. And they're also, the droplets are small. So that means the bigger droplets settle down due to gravity, but the small droplets can travel a great distance. And with this, you know how much is the viral load is of the order of millions, and the infection dose is roughly of the order of this many copies. So you can imagine that how quickly, if you are in the same room as a person who is infected and he sneezes or coughs, that it is very easy to catch this virus. And so the droplet in a single cough or a sneeze may contain as many as few 100 million virus particles, and they travel very fast, and especially the smaller particles travel farther distance and eventually could infect a person. So given the high viral load, the aerosol transmission of small particles, that means roughly about 5 microns or so, is a significant source of infection for this COVID. Now, here is an important thing that you need to ask. So we have a high virus load, and a person who is infected are having this potential of this virus. When the person coughs or sneezes or whatever, a large number of virus particles can travel sufficient distance, and somebody can easily catch 1,000 or so of these virus particles by getting exposed to them. But then the question to ask is, when it's aerosol borne, what really happens in terms of which ones travel, in terms of what is the potential of which type of particles or what particle distribution is important. So if you look at the influenza, you can see the bigger particles, roughly of the order of 50 or greater than 50, they will immediately settle to the ground because of gravity very, very quickly. But if you look at the smaller particles, roughly of the order of, say, less than 5 microns, they last for a longer period of time. In comparison to it, the CoV-1 is again the same story. Bigger particles settle down, or they don't, travel as much, but the smaller particles, especially of the order of about 3 microns, 1 to 3 microns, they remain suspended almost indefinitely. And if you look at the CoV-2, based on the half-life, it's from the risk analysis paper, and you can see, again, less than 5 microns, they can last for many hours, few hours under 80% relative humidity. And here, this paper shows another important result as well, that the relative humidity in the ambience also plays a very important role. Suppose if it is a very dry ambient, then these particles can be suspended in air for a length of time, for several tens of hours. So now, if we then look at the high viral load and the potential for transmission and capturing the transmission in a healthy patient, and the suspension in air of the smaller particles for hours of time. So all these are potential risks for somebody getting exposed. And then if you look at the aerosol stability, this is looking at CoV-1 and CoV-2 comparison. And this is again from the same reference that Kuchipudi also mentioned. But what is important here to note is these aerosols that was used for this measurement, they were generated with the use of a 3-jet collision nebulizer and fed into a Goldberg drum to create the aerosol environment at 65% relative humidity. The reason why this is important, because this is not a clinical condition. So this is purely a laboratory condition, and that's where they have measured this data and reported. So you can't really translate it to the clinical conditions, but nevertheless, they serve as a benchmark, right, to understand what goes on. And if you look at CoV-2, like aerosol we already talked about, if you look at plastic and stainless steel, they can stick onto those surfaces for a very, very long time, for a couple of days. And it is somewhat similar for the CoV-2 also. There's not much of a difference. But then if you look at cardboard and particularly copper, then the time is considerably reduced. And in the case of CoV-2, the copper is very similar. It lasts only for 4 hours, but compared to CoV-1 on the cardboard, it lasts longer. And these are all the caveats in this experimental data, which you can look at the paper for further clarification. So anyway, based on all these factors, high viral load and the potential for transmission through aerosol and the potential for sticking onto surfaces, based on these, the World Health Organization, they made an assertion that the transmission of CoV-2, the virus, is the primary transmission is through droplets, direct contact. So that is in contact with a person who coughs, talks, sneezes, etc. And there is a very high potential for capturing this, contacting this virus. But then there is an indirect contact as well with the contaminated surfaces in the immediate environment, or with the objects used on the infected person, like in a hospital setting, stethoscopes or thermometers, so forth. So therefore, there are 2 mechanisms of transmission, and both need to be looked at very carefully, and how we can prevent the spread of the virus, and especially in the future viruses that could happen. So now let me quickly.
Swami, we have 2 minutes left.
2 minutes, okay. Thank you, Mauricio. So I'm going to do is, on the sensing, there are the previous speakers already talked about many different types of sensing. The one thing that I want to point out here, the RT-PCR is an expensive instrument. False negatives are possible. We need a cheaper pre-screen, for pre-screen purposes, a cheaper point of care sensing. There are photonic devices, label-free capture, which Mauricio will talk about using carbon nanotubes. We have electronic devices, and we already heard Professor Zhang talking about that. But what is important, so I will quickly move these animation. This is the point that I want to stress, and I hope I convince this community enough that it's okay to talk about sensor, but we oftentimes ignore the receiver operating characteristics under clinical conditions so that we can assess realistically the positive predictive value and the negative predictive value in real-time applications. This is one drawback in the current situation, and this has to be circumvented. So in the time I left, I talk about antiviral approaches. In antiviral approaches, the data that how long a virus lasts, we saw that both CoV-1 and CoV-2, the copper surfaces, it lasts for the minimum time, so like a few hours, like 4 hours or so. And in this case, copper and silver have had, it's been known, very well known, that they have antimicrobial properties. And so therefore, this will be very interesting to see. And people have done, using copper oxide containing respiratory face mask for influenza versus actually as back as 2010. And a recent paper in Advanced Material Interfaces shows that copper with a laser surface texturing, so a hierarchical mesopore structure, it rejects bacteria, and very likely it is likely to reject viruses as well. And silver is another very interesting metal with antiviral activity. Silver are gold nanoparticles, especially silver has been shown that it is very good antiviral properties against the African swine fever virus by directly binding to the virus. Okay, these metal nanoparticles directly bind. For the carbon community, graphene and graphene oxide both have been shown to be good antiviral properties. Functionalized nanographene sheets are very good against the HSV1, which is the herpes virus. Both graphene oxide sheets and graphene oxide sheets with the silver particles again have very good antiviral properties. And recently, curcumin is a polyphenol compound, has very good antioxidant, antiviral, anti-inflammatory, anti-cancer, etc. People have tried antiviral cationic carbon dots based on curcumin and seems to have very good antiviral responses to response of antiviral nature. And this could be again something that could be tried. So I talked about these things. Let me summarize my talk. The three main points to take away. Of course, this COVID-19 disease due to this coronavirus 2, it has created an unprecedented large-scale global pandemic, and the world was not well prepared. And the second bullet point I want to emphasize is the understanding is continually evolving. Still, a lot of things are unknown about how this virus is transmitted. Professor Kuchipudi talked about how the virus came about, but then once the virus is in the human body, how it gets transmitted from person to person in direct contact or indirect contact, this information is still evolving. So there are challenges in combating the current pandemic, but I think this is the opportunity for us, for scientists, to look for ways to be better prepared for future outbreaks of similar viruses, coming up with sensors which are more reliable, and coming up with the antiviral approaches to mitigate the propagation, and also better public health policy and public health education. So with that, I thank you again, the Carbon Society, for this opportunity to share with you basically what I have learned and what I'm still learning. But as I said, I'm not an expert, but I hope there's more opportunities to come up with new ideas. Thank you.
Thank you very much, Swami. So I would like to thank Suresh for answering some of the questions, and I will encourage all the speakers to answer the questions from the chat. So you can click on the chat and answer the questions. I think there are no questions for Swami right now. So you want to?
Hello?
Yeah. Do you
Hello?
have? Yeah. Is there a question? Okay.
Yeah, there is a question. This is Chaim Grebel from NGIT.
Only. Yeah. Try to be.
Hi, Swami. Great talk. Great summary.
Thank you.
Could you elaborate a little bit about the photonic device, tagged versus non-tagged systems like IR, Raman, colorimetric, and so on? Just in few sentences. And what do you view their place in view of the other biological systems?
So the question is in terms of how the photonic devices will be used against virus detection. You know, the photonic devices, people have also reported microfluidic devices and using gold nanoparticles and so on, looking for, again, because of a refractive index change, shift in the frequency of whatever the mode that they are detecting. So it could be very high sensitivity. But with any type of sensing, which is what I was trying to say, is in laboratory conditions, they all look great and, you know, great results we get, but people need to exercise them somehow under clinical conditions, and the repeatability of the measurements, false positive, false negatives, and also the aging characteristics of these sensors. That's also something which is not known. So if you want to replace a cheap point of care sensing in the place of an RT-PCR or something like that, it has to be very robust.
Thank you.
Thank you. Yeah, I think we have to move on. So if you have more questions, you can put them in the chat and I will ask Swami to answer them.
Yeah, definitely. Thank you. Thank you again.